There's good reason why we don't eat raw chicken and wash our hands after touching raw meat. While most of you are familiar with Salmonella enterica, there's a potentially more significant microbe we should be worrying about. This bacterium here. This is Campylobacter jejunum, or Campy for short. Campy resides as a commensal in chicken guts, and in humans is one of the most common culprits of gastroenteritis and other diarrheal diseases. While you most likely get symptoms like diarrhea and vomiting, if you ingest enough of this guy, infections can lead to complications like Guillain-Barré syndrome and Miller-Fisher syndrome. Last year in Australia, there were more cases of Campy-related food poisoning than for the other, more infamous pathogen, Salmonella. Just for reference, this rate in Australia is approximately 10 times higher than that of the USA. It also costs the US up to $2.9 billion each year. So what makes Campy so good at surviving in chicken guts? And by extension, what enables it to cause disease in humans? Let's find out. I'm heading into the Cordwell lab at the Charles Perkins Centre in CapA, Sydney. Their speciality is investigating disease processes using a proteomics-based approach and mass spec. Currently, they're investigating various proteins that may be involved in C. jejuni virulence. The one I'm focusing on is an autotransporter protein nicknamed CapA. It's been demonstrated to play a role in adherence and invasion of human epithelial cells. But the results are inconclusive. Why? Because so far, only 2 papers have investigated CapA, and both of them had contradictory results. We basically want to find out if CapA plays a role in colonizing chickens, and also whether it plays a role in causing virulence in humans. We can figure this out by seeing whether a mutant without the gene encoding for CapA expresses virulence phenotypes in vitro. If CapA is a significant player in causing disease, then presumably the CapA mutant will express those phenotypes to a lesser extent. In this study, we looked at 4 in vitro features associated with virulence: motility, antibiotic resistance, biofilm formation, and adherence and invasion of human epithelial cells. We used a hypermotile strain of C. jejuni. Stocks were grown in BHI and yeast media, along with TMP to select for C. jejuni and kanamycin to select for the C. jejuni mutants. Motility. We inoculated 12 semi-solid agar plates with mutant and wild-type colonies, and then compared the distance wild-type and mutant colonies had spread from the inoculation point. We found that mutant colonies traveled much further than wild-type colonies, suggesting that the CapA may play a role in increasing motility. Antibiotic resistance. Resistance of C. jejuni to polymyxin B was quantified in terms of minimum inhibitory concentration via the E-test method. We found that, like for the motility results, the wild-type strain was more resistant to polymyxin B than the mutant, inferring that CapA has some role in antibiotic resistance. Biofilm formation. To compare the extent of biofilm formation between wild-type and mutant strains, we cultured each strain in wells containing either BHI broth or BHI broth plus chicken juice. The chicken juice was to mimic favourable conditions for biofilm formation in chicken guts. 2 replicants were also shaken to model conditions under stress, and the other 2 were incubated without shaking. After incubation, non-adherent cells were washed off with PBS, and the remaining adherent cells were resuspended in broth and dye. After more incubation, all the media was removed so that only dyed biofilm-forming cells remained. We dissolved these with acetone, transferred every replicate onto 196 well plate, and measured the absorbance of each well against a negative control for each condition. Now remember that there were 2 variables we tested. The first was biofilm formation in the presence or absence of chicken juice. So in the first model, which we can think of as a control, in that it is not under stress and it doesn't contain chicken juice, the CapA mutant was better able to form biofilm than the wild-type strain. We can see that in the chicken juice, however, the CapA mutant is less able to form biofilm compared to the wild-type. So from this, we can infer the CapA plays a role in biofilm formation only when colonizing a chicken gut, or at least an environment that mimics one. The second variable was stress, modeled by the shaken plates. Let's look at the non-shaken chicken juice model. Like the control model, there was less biofilm-forming mutant bacteria than the wild-type. We can see a similar trend for the shaken chicken juice model. But if we compare it to the control column, there is a bigger decrease in the proportion of biofilm-forming mutants compared to the wild-type. Could this mean that stress is a factor that upregulates the expression of CapA? Maybe. And there are papers that have looked at CapA's phase variability. That is, how C. jejuni can have different phenotypic outcomes that occur under different environmental conditions, though none of them have characterized it yet. Unfortunately, it's hard to get a conclusive comparison across the 2 different models we used in this study. We would need to study the direct expression of CapA under different environmental conditions, for example using qPCR or MALDI-TOF. And it would be a good idea to conduct this kind of in vitro study before we go infecting a bunch of chickens, or humans for that matter, for an in vivo study. What we can conclude, though, is that in a model chicken gut environment, there is a reduction of biofilm-forming mutants compared to wild type, whereas this isn't the case outside such an environment. Adherence invasion. To model pathology on human cells in vitro, human epithelial cells were grown in 24 well plates in this pretty pink medium. Once they reached 80 to 90% confluency, we infected the cells with colonies of wild type and mutant strains grown on BHI plates. After 4 hours of incubation, we washed off the non-adherent bacteria with PBS and lysed the cells in one of the plates with a detergent. What's left are bacteria that have adhered and invaded, and serial dilutions of these guys were plated onto 5 BHI plates, and then the number of colony-forming units were counted. Now, to figure out the proportion of bacteria that invaded, we added gentamicin to the other plate of human cells to kill off only adherent bacteria. After another 2-hour incubation, we lysed the cells with detergent and did the same thing as before. Now, to our surprise, we found that more CapA mutants had adhered and invaded the human cells than the wild type, which is the opposite of the pattern observed in all previous studies. While the differences were non-significant for the adherence and invasion assay, they were significant for the invasion-only assay, and the trend was still the same. So CapA doesn't seem to increase the invasive ability of Campy. The most reasonable explanation for this would be the phase variability of the CapA protein. The Flanagan paper concluded that CapA isn't always needed for chicken colonization, and that the Campy isolated from chickens completely lack the CapA gene. Putting all of these results together, we can see that, except for adhesion and invasion, the wild-type strain exhibits virulence phenotypes to a significantly greater degree than the CapA mutant. Those adherence and invasion results also tell us that the behavior of CapA is much more complex than we originally thought. There's a lot more factors to consider before we move on to in vivo studies targeting CapA, like what physiological and environmental conditions will induce the expression of that gene, or even make it necessary for that gene to stick around in the Campy genome at all. All this is a bit daunting when you think about the thousands of other proteins that have been characterized in Campy colonization, but figuring out what this tiny protein really exists for will help us develop a new target for preventing Campy infections, characterize the phase variability of proteins involved in other diseases, and approach research with a greater appreciation of just how complex host-microbe interactions can be.